you. I'm excited to introduce Dr. Brandie Quarles-Chidyagwai. She's our speaker today. Brandie got her bachelor's at University of Virginia in biology, where she worked with Deborah Roach, working on that really cool Plantago lanceolata system and understanding aging and trait expression, how trait expression changes through age in that kind of, if I can call it a model system for aging and senescence in plants. And then Brandie went on to get a PhD at Duke with Kathleen Donahue, working in integrating ecological and evolutionary responses to climate change, focusing on the role of dormancy and dispersal of seeds and how they mediate those responses, and also thinking about evolution in relation to those traits. And then we were lucky enough to get her to join our group as part of our integrated biology project starting in 2023, where she's been studying ecological and evolutionary responses to climate change in what we think is this very cool but also very complicated wildflower, Streptanthus tortuosus, the mountain jewel flower, and really integrating these kind of demographic components as well as genetic and evolutionary responses, which seems to be right in Brandie's wheelhouse, and she'll be telling you more about that today. And in addition to really excellent research experience and accomplishments, Brandie is also a really excellent teacher and mentor, and just a really wonderful community member that we're delighted to have in our labs, actually in my lab and Jewel and Lou's lab, and as part of this community. So I'll let Brandie take it away from there. Thank you.
Thanks for coming, everyone. Yeah, today we'll be talking about some of the work I've been doing in Jenny and Julian's labs, focusing on adaptational lag and potential for gene flow from lower elevations to rescue high elevations. No, it worked just before. Oh, there we go. Okay. Yeah, so we know that climate change is impacting the environment globally, and if we focus on high elevations, we're seeing warmer temperatures and reduced snowpack. This is actually a picture from the Palisades Tahoe Resort this past December, and so we're seeing those changes right before our eyes, unfortunately. In addition to temperature changes and snowpack changes, we're also seeing just shifting in the timing of our seasons, where our springs are coming earlier, and our summers are staying hotter for longer into the fall. And so what can plants do to respond to these changes? They can't just get up and find a cooler location to go to or a shady spot to move to once they've already started to germinate. And so they can either adapt genetically to those changes, disperse spatially through either their seeds or their pollen, or sadly go extinct. And of course, there's lots of complexity built into this, but this is a useful framework to sort of think about these processes from. And if we think about a plant that's currently growing at high elevation and what it might do after climate change, one option is that it might be able to respond to those changes and still continue to live at those high elevations. Maybe it has sufficient genetic variation. The selection is acting quickly enough and fast enough for it to get there and do what it needs to do. Or it won't. And so there's also a sign that we could be seeing adaptational lag, where they're just not able to keep up with these changes in the climate. And then if we now think about low plants that are currently growing at low elevation, what might happen after climate change is that they might be able to move upslope because they're pre-adapted to these warmer temperatures that higher elevations are starting to get. And so this higher elevation environment might be somewhat similar to what they're used to down at lower elevations. And there have been some previous evidence for adaptational lag in high elevations and potential for upslope migration. And so this is in Boca stricta, which is a plant that's native to the Rocky Mountains. So basically here, I'm just pulling one of their figures where on the x-axis is elevational difference. So they did these reciprocal transplants across elevation. And so on the left side of this x-axis means that they took a low elevation plant and moved it up to a higher elevation. On the right side of this axis, they moved a higher elevation plant a little bit lower down in elevation. And then on the y-axis is the probability of reproduction. And they had two treatments here. One was just regular ambient temperatures or conditions, and the other one, they removed snow sort of in late winter. The main thing I want you to first think about is this zero elevation difference. That means the plants stayed at their home elevation. And this is not the highest point on this y-axis, right? So if we expected local adaptation, then at 0, we should see the highest probability of reproduction. And that's not what we see. So that's some evidence of some adaptational lag going on in this system. And then also you can see that the plants that were from lower elevations that moved up to higher elevation actually had the highest reproduction probability of reproducing, regardless of the snow removal treatment. And so this is some evidence that low elevation plants might be able to survive at these higher elevations. They might be able to help out these local high elevation plants that maybe aren't doing as well in their home sites anymore. But it's not that simple in all systems, especially in Mediterranean climates like we have here in California, in the Central Valley and the Sierras. Here in low elevations, we get these hot, dry summers. And so plants tend to have to wait to fall rains to germinate. And so they have this sort of winter through spring growth season. And at high elevations, they have to contend with high snowpack usually for most of the year. Historically, they've had high snowpack. And so this means that they usually had to wait until spring snow melt in order to germinate. And so their growth seasons were really constricted to just the summer months. So these plants are growing at very different times of year and under very different conditions they're growing. In addition, because of these short growth seasons at high elevation, a lot of plants have to do this biennial life cycle where they don't reproduce until their second year. Whereas low elevation, they can kind of choose between an annual or biennial life cycle in some systems. Okay, so if we think about how this sort of seasonal differences and life history differences might impact these dynamics, we think about performance in a hot summer and we're at high elevation. So we take a high elevation plant, we keep it at high elevation. What might be happening as these summers are getting warmer? And so we might see that these seedlings have poor survival into adulthood because they're not adapted to these hot, dry summers that they're now experiencing. But if we want to be a little more hopeful, maybe we say they're somehow able to keep going and maybe it's just a slow process of selection is happening and maybe they're not growing perfectly, but they're still hanging around. And then if we consider we take a low elevation plant and move it to high elevation, then maybe those plants are used to those hot, dry summers or those hotter temperatures in general. And so they might be able to survive quite well into adult plants. But then we have to also think about the winter. And so in a winter, if a high elevation plant is used to the winter, so it knows to germinate after the snow melts, not going to germinate early, hopefully. And so those plants are more likely to actually successfully turn into seedlings. And then let's say we're in the optimistic mindset, and so it's able to somehow survive those summers, and then it's used to those winters as well, so it's able to survive into its second year. It's just not doing great, but it's still able to hang on because it knows how to handle those winters. And then if we consider the case of our low elevation plant moving to high elevation, then these plants may not know that if they get snow or a little bit of early cold precipitation at high elevation, that that's not time to germinate. And so maybe they germinate right before snow comes, which wouldn't be a good idea for a little seedling. And so there's a case that those seedlings just don't make it and die. Maybe some of them get lucky. And so the ones that maybe get lucky are able to survive to adult plant, but then because they have these short growth seasons, they probably aren't gonna have time to reproduce in their first year. And so surviving that second winter is really important, and maybe they just don't have the adaptations to make it to the second year. So this sort of seasonal variation can play a big role into what we might expect to happen in this system. Okay, so maybe the solution is to make a cross between the two. Maybe there's gene flow might be the answer. And so if we take these high elevation plants and cross them to some low elevation plants, we get these F1s, we grow those at high elevation. Maybe these are the ideal plant. They're able to germinate at the right time. They're able to survive those hotter summers well, and survive into their second year of life well. And so maybe these F1 offspring of either natural gene flow that happens up elevation through pollen or seeds, or through assisted gene flow, which would be human sort of conservation aimed gene flow, sort of help these high elevation plants respond to these changes they're experiencing. So that's something that we're interested in. And so some big questions in the field right now are, how will plants growing at high elevations respond to climate change? So given these changes high elevations are experiencing across the mountains everywhere, what are these plants going to be able to do? And if there are signs that those populations are struggling, is there a possibility from gene flow from lower elevations to rescue high elevation populations? And specifically, my research questions have been, is there signs of local adaptation in these high elevation plants? Or are they showing signs of adaptational lag in response to these recent climate changes? And sort of correspondingly, do plants from lower elevations survive better than local plants at high elevation? Or is there sort of this seasonal variation that's playing a factor into these patterns? And so maybe plants from hotter origins survive summers better at high elevation, but the plants from cooler origins survive winters better. And so maybe there's some of that going on. And then finally, sort of what role might gene flow be able to play in all of these dynamics? Will gene flow between low and high elevations increase fitness, or is it just going to end up in a disaster? To answer these questions, I've been working with Streptanthus tortuosus, the mountain jewel flower, has these beautiful jewel flowers. So it's a really awesome plant to work with, especially when it's flowering. And so we have populations that grow across a gradient of latitude and elevation. And so here's some of the populations we included in our study. Each dot is colored by elevation. So we have a good representation across latitude and elevation. And because of these differences in elevation, they do experiencing these vast seasonal differences across the range. So very similar to sort of the background I set up. And because of these differences in seasons, these plants also grow at very different times of year, as I sort of talked about. Okay, and we also have some evidence of actual climate change at these sites. So this is a PCA of many different climate variables. And on PC1, it's sort of like a cold, snow, warm, no snow gradient across the x-axis. And PC2 is a dry to wet axis, mostly wet in colder months, so not wet year round. And each of these points is one of our populations, and the arrows point from historical climate to recent climate. And so we can see that almost all of these arrows are pointing from left to right. And so these populations are getting warmer and getting less snow. And a lot of them are also pointing upwards. And so they're getting wetter, which might seem a little strange, but again, they're getting wetter in colder months. They're not getting wetter year round. And we know that with climate change, there's been previous studies in California that show that we may not just get drier overall, but maybe just the patterns of precipitation are going to change. And so that's sort of reflected here. The next thing to notice is that these higher elevation arrows are a little bit longer than these low elevation arrows. So our high elevations do seem to be changing in climate a little bit more than our low elevation populations. And then the final thing from this figure is just, I pointed out our 2 years of our experiment on here. 2023, I don't know if you guys remember, it was a very wet year here. They got a lot of snow. So it was a little bit, I don't know, off in its own world in the climate space. But then 2024 was a little bit more of a normal climate year. Okay, so first we're going to address these first 2 questions of what happens if you put high and low elevation plants at high elevation, how are they going to grow? And so for this, we conducted a common garden at high elevation. And so this is the site we conducted it at here, pointed with the arrow. And we grew 23 populations from across the range. And we compared their home climate for the past 30 years to the garden climate that they actually grew in. This is a picture of our garden up at Wright's Lake. And these are some of our plants growing. And then to get at this sort of like, are we seeing changes across, due to these sort of seasonal differences the plants are used to growing in, I measured fitness across the life cycle. So we germinated the plants in the CEF right downstairs, and we let them grow for 5 weeks, and then we transplanted them into the field. 3 weeks later, we measured establishment. And then if they survive to budding or not. And so these are tiny little buds they make, which is just the initiation of reproduction in this species. And then how many fruits do they make? And so these are the fruit pods that they make there. In addition, we measured first year survival. So either you survive the first year by successfully reproducing in the first year, or you just survived as a vegetative plant. There'd be 2 ways to do that. And then we measured winter survival. So survival from October, when last time we were able to get up there, until as soon as the roads opened and we can measure winter survival. And then also survival to reproduction in their second year, and then fruit number in their second year. So sort of trying to capture all the different life stages. Okay. And then we also had 2 measures of reproductive fitness. One, whether or not you produce fruits or not, so the probability of reproducing, and then total fruit output. Okay, and then I mentioned climate distance, and so we had 2 metrics of climate distance. The first was temperature distance, and so this is the temperature of the home site's past 30-year average minus the garden site, depending on if it's a first-year fitness metric, it was using the 2023 temperature, and then if it's second year, 2024. And then we also had a more general climate distance. And so this included temperature, precipitation, snowpack, and 12 other climate variables. This is an absolute difference and also sort of like a cumulative or multivariate distance. So all of our results are going to have either temperature distance or general climate distance on the x-axis, and then one of the fitness components on the y. And so for temperature, anything to the left of 0 is going to mean the home site was colder than the garden. Anything to the right of 0 means the home site was warmer than the garden. So if we see a positive relationship, that means that sites from warmer climates perform better at the garden. If we see a negative relationship, that means that sites from colder environments in the garden survived or reproduced or whatever the fitness component is better. And finally, if we see the highest fitness at sort of 0 climate distance, that means that there's signs for local adaptation. And very similar with the general climate distance, it's just that the farther away from 0 means that the farther away the climate generally is from the garden. And so positive relationships means far away climate is better, and negative means the closer climate is better. Okay, so first we're going to look at establishments. Remember, this is measured 3 weeks after we transplanted them in the field. I have temperature distance on the x-axis here. And our colors are still the same, so that warm is low elevation, purples and blues are high elevation. And we see this positive relationship, just like I said before. And so that means that populations from warmer home sites established better in the garden than other populations. And this is mostly driven by these low elevation populations that had higher establishment. And I've pointed out our local populations. This is the population that actually grows at the garden site. And you can see it had decent establishment, but not as high as some of the low elevation populations. Okay. And then next, if we look at year 1 survival, again with temperature distance on the x-axis, we see another positive relationship, which again is these low elevation populations from warmer home sites that had higher survival to the end of the first year. And again, our local population is not doing terrible, but not doing the best, so not showing signs of local adaptation per se. And we had a few mid-elevation populations that sort of did something different. Not sure about those. Okay, and then if we look at winter survival, so for winter survival, we didn't see any significant relationship with temperature distance, but if we looked at the general climate distance that included all those other climate variables, we did see a positive relationship here. And so again, it's sort of like our lower elevation populations that were from climates generally farther away than the garden site had higher survival over the winter. And this is a little confusing. We really anticipated that that's where the high elevation plants might be able to have the advantage because they're used to growing at these high elevation winters. But the low elevation plants were able to pull it off somehow. I have some ideas of that if anyone is curious, you can ask me later. But then when we move into the second year, our patterns sort of shift. And so now we have temperature distance again, but survival to reproduction in the second year, we have this negative relationship. And of course, this is contingent on you actually making it to the second year. So that's why we have less points on this graph. And we only had 2 high elevation populations make it to this stage. But those 2 were able to survive to reproduction much better than these low elevation populations from warmer home sites. So really it's the local population and 1 higher elevation population from a colder site that were able to have quite well survival in their second year. But this actually didn't end up making a big difference on overall reproductive fitness. And so here I'm showing you probability of reproducing. So that's, did you make any fruits or not? And temperature distance again on the x-axis. And we have this positive relationship that is maintained here. And so those low elevation plants that were able to survive through the first year and over the winter well, that made a huge difference on them being able to even make a fruit or not. And our local population is not doing quite well overall if you look at this fitness metric. And this is also represented in our total fruit output as well. While this relationship was not significant, but we do see this sort of positive trend where even though these high elevation populations were able to survive to reproduction better in their second year, because they had a hard time even getting to the second year, they weren't able to have higher reproductive fitness overall. Okay, so some takeaways from these results. If we're asking our question of our local high elevation populations showing signs of adaptational lag, it does seem like they may be at risk of population decline because we are seeing that they're having poor survival through the first year and they did not survive that winter very well, unfortunately. But a couple of them made it. So, not completely terrible. And then will plants from hotter origins survive summers better at high elevation? We do see that the plants from hotter origins, which is mostly the low elevation plants, survive the first summer and the first winter better. So they did a good job at that. But if we just take these low elevation plants and move them up to high elevation, which in the conservation world is called assisted migration, it might not be super successful because we did see that those higher elevation populations that made it to the second year were able to survive to reproduction better. And so this may be a case where assisted gene flow, actually crossing these plants, might be a better conservation strategy, where we take whatever genes helped these low elevation plants survive that first summer and first winter better, and combine them with whatever genes help those high elevation plants survive in their second year to reproduction. And maybe we get the ideal F1 cross that can survive in these higher elevations. So that brings me to the second part of this talk, which is exploring the fitness of crosses between plants from different elevations. Okay, so here we perform crosses between the local population, so this high elevation, right, it's like 2 population, and populations from low, mid, and high elevations. And so we took a low elevation plant, crossed it to that local high elevation plant, mid elevation cross, and then another high elevation populations that were not the local population crossed to it. So we had 2 donor populations from each elevation, which I'll show you what they are, which might make more sense in the next slide. And then we had about 1 to 5 unique crosses between each donor. It just varies based on which crosses were successful and survived to be able to actually be transplanted to the field. And so here are the populations that we crossed to this local high elevation population. And so did spread across latitudes still. And we grew these F1s from these crosses at the same high elevation garden. So at the same local site as the local right site 2 population. And we compared the donor climate, so the climate of these non-Rice Lake 2 parents in the crosses, to the garden climate of 2024. These plants were planted in 2024. And we measured the same fitness metrics. Okay, so if you start again at establishment, which again is 3 weeks after transplant, this is general climate distance of the donor. And our points here are the triangle. This is our local population just for reference. And then all the circles are our crosses. And each of the crosses is, so this would be one of the high elevation plants crossed to this local population. And here we're seeing that there's not anything clearly popping out with relation to the climate of the donor. We don't see any super clear trends here, or not any super clear trends in terms of elevation of the donor either. But generally, the crosses had higher establishment than the local population. So one thing we're seeing. And if we look at year 1 survival, so after you established, did you survive to the end of the first year? We see, oh, did I skip the slide? I might have skipped the slide. Oh, I did. Okay, I'm gonna back up first. So now I'm showing you geographic distance of the donor. So I was curious, like if we're not seeing climate have any relationship with fitness, maybe it's the plants that are, or populations that are closer to that local population that might have better success in the cross generation. Just because maybe if they're closer, they have had more recent genetic exchange between the two. And we do actually see that with the geographic distance of the donor, there is a positive trend. I'm still working on this analysis, so I don't know if it's significant statistically yet, but it does seem like the populations from farther away geographically, when crossed to that local high elevation plant, had higher establishment, which is a little bit confusing because you expect, my expectation was that the closer you are, the better you would do, and it's not what we're seeing. Okay, so now I'm moving to year 1 survival. And then, oh, I forgot to tell you, these are sample sizes. And so this, we had 29 Rice Lake 2 plants that made it to this stage. Yeah, so that's what those numbers are, just for reference. And again, we have climate distance of the donor on the x-axis. And here, now we're just getting a variation of populations crossed Rice Lake 2 that are doing better than others. So for example, if we look at, oops. Okay, anyway, if we look at the two of the high elevation populations that we crossed to this local population, one of them did well and the other one, when crossed to Rice Lake, completely did not make it to the end of the first year. And so we have some of those populations are having 0 survival when crossed to Rice Lake, and some of them are surviving. And then if we look at this one with geographic distance, we also see a variation of patterns here. And so if we look at these two, well, these 4 really. This, for example, if you take this low elevation population that was crossed to Rice Lake, the one that was closer to Rice Lake than the population that was further away was the one that had higher survival. But then if we look at the mid-elevation crosses, it's the mid-elevation cross that was population that was further away geographically that had higher survival than the one that was closer geographically. So it's, now we're getting some variation in that some cases being closer maybe is better, in some cases being further away is better. Okay, if we move to reproductive fitness, so did you make a fruit or not? Then we're still seeing very similar trends where we have this variation of which crosses or which populations cross Rice Lake are doing better. And we look at geographic distance and probability of reproducing. We're starting to see a little bit of positive trend again, but mostly it's this mid-elevation cross that just seems to be doing well. And that becomes even more apparent when we look at the total fruit output. And so here is where this mid-elevation population when crossed to Rice Lake had really high fruit output and was able to do better than other populations across to it. And it was able to do better than the actual local population itself. And then if you look at this with geographic distance, that mid-elevation population again is also the one that was furthest away geographically. So both mid elevation and farthest donor seems to be doing the best. So from these results, it seems that gene flow for mid elevations might be the best option. And the furthest population geographically contribute to the highest fruit output, which is a little bit counter to expectations going into this. And some populations just don't seem to be good sources of gene flow, and some seem to be better. So, you know, each of our kind of pairs of high, low, and mid elevation populations, one of those completely did not survive the first year, and the other ones were able to make it to further life stages. So can't just say, I'm going to pick any mid elevation population and cross it to the local population and it'll do well. It really depends on the specific population that you're taking plants from. But wait, there is variation across years. So we repeated this experiment again in 2025, and we see different results. So again, on the left side is the results we've already seen of year one survival and geographic distance. And I'm showing you the same results from plants we planted in 2025. And first of all, you can see that these populations that did not have survival in 2024, they had a greater than 0 survival in 2025. And to give you an easier way to look at this, I made this into a reaction norm. And so here we have 2024 and 2025. Each of the points is one of these crosses to Wright's Lake. And we just see that there are big changes in some cases in fitness between years. And so, for example, this one high elevation population that crossed Wright's Lake had 0 survival, and in 2024 and 2025, it had 100% survival. So that's a pretty big difference. And so if we were doing this experiment just in 2025, we would say, oh, this high elevation population must be the best one to cross to the local one to help it do well. But if we had done it just in 2024, we would have had a very different conclusion. But there are some cases where things are sort of staying consistent. And so here's this low elevation population that across Wright's Lake did decently in terms of year one survival in both 2024 and 2025. And so there are some cases where even if who's the winner or loser is changing, some populations are sort of staying consistent across years. And remember, this is just year one survival. And so when we looked at probability of reproduction and sort of total fruit output, this mid elevation population sort of started to take the lead. And because these plants don't reproduce in their first year, I don't have the answer on that yet for the 2025 plants. So it'll be interesting to see in this upcoming summer, whether or not this mid elevation populations, or even if this high elevation population that had 100% survival actually is able to have higher reproductive fitness as well. So we'll find out soon. And so from this, if we were trying to approach this from a sort of like, what's the best strategy for assisted gene flow or for saving high elevation populations that are struggling, it might seem that maybe taking multiple sources for the gene flow might be the best option, because we're seeing this yearly variation in fitness, and that yearly variation, I don't know for sure, but I'm thinking it's probably just this big variation in climate that we see between years. And so I think, like, you know, sort of hedging your bets and sort of getting a mix of populations might be best. Or you could focus on the more consistent sources. And so the ones that are sort of having good survival and reproduction each year might be a sort of a safe option. And so currently, we're still working on evaluating the potential of assisted gene flow in the field. And so we're going to plant more crosses in the field this upcoming year, and then measure the reproduction of the plants we planted in 2025. And in addition to F1s, we also have backcross 1s and backcross 2 plants that will show like further generations of gene flow. How would those plants survive? Yeah, and I think just continue to do this. I'm really glad we've done this in multiple years, and we're going to do it another year, because we are seeing this change in fitness across years. And so really curious to see what's going to happen this upcoming year, especially with the snow dynamics that we've experienced this year. And then it'd be really great, because of all this variation, to have some sort of quantitative framework to sort of make better predictions about who's going to be the best source of gene flow or which populations are going to be successful at higher elevations. And so we're working on integrating demography and genetics into an integral projection model where we can sort of explore the consequences of assisted gene flow and incorporating this climate variation into there to try to make better predictions about how populations are going to do and what happens if you start crossing plants together. And with that, I have a lot of people to thank, especially all the undergrad students. These are all the students that helped to collect the data that went into this talk. It takes a lot of work. We go up there every week in the summer to collect this data. And all of my advisors and people in the lab who have helped out. And I'll put up my research questions again as a reminder of what we found and take any questions.
Yeah, that was a super cool talk. And I was just thinking, seeing like the high elevation plants like do horrible at, the common garden site, and then seeing the year-to-year variation of some doing well and some not. I was wondering, has there been any observations of like herbivory pressure? Like I know there's a lot of variation in insect outbreaks each year. And I was wondering, are the lower elevations more protected, or are there different pests coming in different years, or just kind of throwing the herbivory side into the ring? If you have any thoughts.
That's a great question. We do measure like sort of like, is there signs of herbivory, yes or no, every year? And then last year we started doing a little bit more of a fine tune, like what's the damage? I haven't gotten into the damage data yet, but just from like whether or not we're seeing herbivory on plants, it does seem pretty consistent across plants from different elevations. So it doesn't seem like the herbivores are picking their local high elevation plants to eat on. They're sort of eating everything. In terms of difference in herbivory pressure between years, anecdotally, I can say I don't remember seeing like especially different herbivores, but that's something that could be at play that, maybe there's coming out when I'm not there the 5 hours a week that we're going up there. So
Awesome.
yeah. Yes.
Yeah, in the maybe you could just tell me, there's an amazing amount of data here. I'm trying to sort of parse through it all. When you sort of do the experiments across 2 years and get through different results, is it's the same populations you're crossing? Is it the same individuals within those populations you're crossing, or do you think there's some possibility that you're just getting different individuals being the dominant parents across those years?
That's a great question. So we generally have this same sort of mix of maternal families or parental families that are going into the crosses. But it's also an outcrossing species. And so there's also just like variation, there's more recombination going on. But I would say that for the most part, we have been using the same parents going into the crosses, but there is some little variation. So yeah, I am going to double check that like the parents are exactly the same, like in that reaction arm, for example.
I guess I'm wondering, like, so if the parents are the same, but like, are the actual sires the same or not?
Yeah.
I mean, do you have any, are you doing any genetics on these to?
We are,
Okay,
yes,
cool.
yeah. So we also, every year we have been sequencing or collecting the DNA before we plant them up there. And so we should be able to get at that question too, just who are the parents genetically, yeah. Yes.
I guess given your results showing that these low elevation populations are doing better and that or perform better under common conditions, and that the F1s are doing better, like I'm kind of surprised that you aren't just seeing more changes or more gene flow in these different populations. And I guess I was wondering, like what you think is really limiting the ability of these populations to adapt and for there to be gene flow.
Yeah, like just natural gene flow.
Exactly.
And
Yeah.
elevations.
Like why do you need to do assisted gene flow? Why can't they just?
Yeah. Well, I think first of all, I think it's like really surprising to me that so we know that this species sort of started in lower elevations and historically migrated upwards in elevation, but it doesn't have great like dispersal advantages. So it seems to sort of generally just drop. I don't think they're great at dispersing in the wind. So it's kind of like a miracle to me that they even made it to high elevation in the first place. So I do think like there's just generally, it's a hard time for them to get there on their own. That's just my general take, but but you're right, because they are doing better. So like if the rare ones do make it up there, why aren't we seeing them, sort of having an impact? So hopefully with the genetic analysis that we're working on, we should be able to see if there has been any gene flow already, maybe. But yeah, it's a great question.
Related to that, I'm wondering if, so the pattern of seeing the ones further away crossing and doing better indicate like that's what outbreeding depression might look like, right? Where they're sort of locally inbred and if they have really poor dispersal ability. Is there any evidence that that's the case, or are they generally really diverse populations? And
Yeah, so so far it does seem like the genetic diversity is pretty similar across the population, just from some of the preliminary genetic analysis that we've done. And so I don't say that I don't think there's clear evidence that that's what's going on here, but it's definitely something we can look into. Yeah. Yes.
Could you talk a little bit about why you think the lower elevation ones are doing better? I've been thinking about, Jenny and I have been talking about the trade-offs between being stress tolerant versus disturbance and colonizing after disturbance with plants. So I'm getting confused. And then also like temperature to me is like that's stress. So I can like see that as like being a stressor. But when we think about snow, I'm also a marine ecologist. So like when we think about snow, do you think about that as like disturbance? Or do you also think about that as stress? Or do you think about that as both? And how does that play into like these plants and how like maybe the southern ones are better at taking up the northern?
Yeah.
Even though they started in the south too. So that's like, I don't know. That's
There's a lot going on here. Yeah.
Yeah.
Yeah, that's a great question. So I think for the snow part, I do think of it like lack of snow as sort of a stressor because the plants, when you have like snowpack, it sort of acts as like a blanket for the plants. And so it sort of insulates them from the even colder temperatures. And so if we're having variability in our snowpack and the plants are getting exposed to the ambient temperatures in the middle of winter, they're experiencing much colder temperatures than they would if they were tucked under the snow. And so I think that lack of snow is probably a really big stress. And that sort of like leads to my current idea of why the low elevation plants might be doing better is because remember I said they tend to germinate after fall rains. And so they are sort of growing as seedlings or young plants through low elevation winters, which are low elevation winters. They're not that cold, but they are cold. And high elevation plants are never really getting to experience super cold temperatures because they're usually under snow and they're growing as plants in the summer. And so I think there might be some differences in cold tolerance between elevations, but I don't have actual evidence of that yet. And let's see, so, and then in terms of like, yeah, temperature as a stress, we actually did measure weekly temperature in 2023 and like the survival and response to those temperatures. And we did see that weeks that were hotter, like a week or two later, we had higher mortality. And so it does seem to be a big stressor for the plants as well. I don't know. So far, I'm thinking the low elevation plants, they can grow taller. So maybe they have more stress tolerance and also just have these like sort of fast growing traits that help them sort of beat some of these stressors maybe. But yeah, these are all just like ideas that I thought of in my head, but haven't actually experimentally tested for sure yet. I also think I do have some evidence that the low elevation plants do have more plasticity in size and phenology, I think. And so maybe just having more plasticity is helping them sort of survive better in these different conditions. That's one idea. Yes, Graham.
So I was following up on Julia's question. It looked like there's heterosis for the F1s generally doing better. And I wonder if you've compared to the parentals from each of the sites as a way of demonstrating heterosis there.
Yes, yeah. So that's something, another analysis that I've just started getting into the crosses analysis. So that is on my to-do list. The tricky thing though is that like our crosses were made in the greenhouse, and all of our populations come from wild collected seed. And so there's like this difference in the maternal environment that I'm not sure like how much that would influence like my ability to like confidently compare the parents to the crosses. But I'll try anyway and see what it is. Yeah. But I do think there is some heterosis happening.
And the back process should help you get at
Yeah, the
that
back
too,
process
right?
should help too.
That's
Yeah.
awesome. Thank you
Thanks,
so
Brandie.
much.
Yeah, thank you.
I'm really good.